right, well, thank you everyone for joining us, and welcome to the Inflammation Pathways session. We are excited to have you here. We have a great, my name is Liao. I'm joined by my co-host Tazeen Ahmad. The speakers are Dr. Jessica Allegretti, Dr. Thorsten Mempel, and Dr. Andy Lester. In the interest of time, we are actually going to jump right in, so I'm going to refer you to the online bios to read about all their impressive bios. So why don't we start off with the first question is asking and getting our panelists to think about how inflammatory treatments that target inflammatory diseases have evolved. We have a lot of great treatments out there. As a rheumatologist, I know we have a lot of targets, but there's also a lot of need. There are a lot of conditions that still are uncontrolled, conditions that don't have treatments. So I'd like to hear from the panelists about new, emerging, exciting pathways that you think will be helpful in the near future. I thought I'd start with Dr. Lester to tell us a little bit about complement.
Okay, I'm Andrew Lester, and Chief of the Division of Rheumatology, Allergy, and Immunology at MGH. And there were two targets were mentioned in the questions that were sent to the panel before, and I'll just introduce both of them. One is the complement pathway, and the other is the FcRn pathway. So complement is one of the oldest parts of the immune system. It evolved to deal with blood-borne pathogens, and it's activated by the bacteria themselves. It activates the complement pathway, which is a number of proteins in the serum that lead to a number of biological outcomes, including the opsonization of the bacteria, which means it gets phagocytosed by the immune cells. It leads to the killing of the bacteria through a membrane attack complex, and it also releases anaphylotoxins like C3 and C5a, which basically is like an alarm bell, and it calls in many immune cells into the site of inflammation. It's an important pathway. There are human deficiencies in complement, which lead to infections. So early in the complement pathway, defects lead to infections with encapsulated bacteria like Neisseria meningitidis. Later defects in complement, like a C1q deficiency, leads to autoimmune diseases like lupus. And one of the early complement deficiencies that actually has a therapy is PNH, which is paroxysmal nocturnal hematuria, which is a deficiency in a protein on a red blood cell that actually inhibits the complement pathway. And those patients develop lysis of the red blood cells. And the therapies that have been out there for a while, a therapy that actually blocks the complement activation, C3 activation, and that's a therapy for PNH, and that was out there in 2000, I think, in 2007. So there's been a lot of interest in the complement pathway, the cascade. It affects a lot of different immune cells, and then there's been a reemergence of their interest in complement with the identification of a molecule called avacopan, which was approved for the treatment of ankylosing, which is a serious disease that doesn't have a lot of therapies. So there's a lot of interest now in trying to figure out what other diseases might be amenable to complement inhibition. And I think we're going to see a lot of clinical trials in that area, a lot of new drugs. And it was sobering for me just following along, because it's taken many years from the identification and even having inhibitors out there for PNH, for instance, to now have a new therapy for ankylosing. So sometimes it's not an easy nut to crack to figure out what's the right drug for what disease, even if you actually have the target and the pathway. And another new one is the Fc receptor, neonatal Fc receptor, which is quite an interesting target. This is a molecule that binds to the Fc portion of IgG and was identified because it actually transports IgG from the mother to the fetus. A very important molecule. It also has another role in that it actually recycles immunoglobulin IgG inside the cells to outside of the cells, and actually preserves the immunoglobulin and leads to a longer half-life. So it's actually been targeted. If you target the Fc receptor, neonatal Fc receptor, actually blood levels of IgG go down. And there was a recent, so there's a lot of interest in doing that to lower pathogenic immunoglobulins for diseases where it's known that there's a pathogenic immunoglobulin, like myasthenia gravis with a drug that blocks the FcRn was recently approved. And I think we'll see a lot more trials and a lot more interest in a target like that, which is a newer target than complement. So we had one old and one new.
Great, thank you, Dr. Lester. And so, you're speaking a lot about, immune-mediated types of targets, but, we've been hearing a lot about cell therapy. So, Dr. Mempel, can you talk to us a little bit about that?
Yeah, so my name is Thorsten Mempel. I'm the Associate Director of the Center for Immunology and Inflammatory Diseases at MGH, and I have a research lab that works on cancer immunotherapy, cancer immunology, including regulatory T cells, but also CAR T cell therapies. And the reason I'm here today is because CAR T cell therapy and Treg therapy have now started to actually emerge as novel therapeutic modalities also in the inflammatory disease space. So in one arm of therapy is CAR T cell therapy as an alternative to antibodies to deplete pathogenic B cells driving autoinflammatory diseases. And here the benefit seems to be that with CAR T cells targeting, for instance, CD19 expressed on B cells, one achieves more profound depletion of B cells that reaches also in tissues, memory B cells, and thereby achieves a more sustained and more complete eradication of pathogenic B cell clones with longer lasting effects than is possible with antibodies. The other modality are regulatory T cells, both polyclonal but also TCR or even CAR engineered regulatory T cells that actively promote tolerance and can thereby re-establish a homeostatic set point in a patient with inflammatory disease that also has the potential to be very sustained in its efficacy and also works very much upstream in the pathogenic cascade that causes inflammatory diseases and has great benefits in that sense.
Great, well, thanks for that wonderful overview. So Tregs are of a lot of interest in inflammatory conditions, including IBD. So Dr. Allegretti can tell us a little bit more about that.
Yeah, absolutely. Thank you so much for having me. My name is Jessica Allegretti. I'm the medical director of the Crohn's and Colitis Center at the Brigham and Women's Hospital. My lab is a clinical trials lab. We work in novel therapeutics for IBD as well as microbiome therapeutics. And so this is an area of real interest right now in IBD. There is a lot of drug development happening in this space, and I think a lot of the known pathways we're really focusing on immune suppression. However, there's been a lot of interest in looking at low-dose IL-2 and actually being able to upregulate regulatory T cells as a therapeutic option in both ulcerative colitis and Crohn's disease. So we know that low-dose IL-2 has already been shown to be efficacious in things like graft versus host disease, which actually can look quite similar to inflammatory bowel disease. And so we actually trialed this initially in ulcerative colitis. Using varying doses of low-dose IL-2, you can actually see expansion of regulatory T cell populations in both the blood as well as the tissue. However, there is a real fine line where you don't want to sort of overshoot and upregulate conventional T cells as well, because then that leads to additional side effect profiles. So this is now also being studied in Crohn's disease, and there are actually several companies right now with IL-2 muteins in development looking at this for more so ulcerative colitis than Crohn's, but that will certainly be coming. But I think, again, thinking about how we can work with the innate immune system as opposed to just suppressing it is really this hot area of development within IBD. And I think along those lines, my real area of interest is how we can use the microbiome, augment the microbiome to, again, really impact some of these really critical pathways. And so FMT, or fecal microbiota transplantation, has really been used quite broadly, of course, for the prevention of recurrent C. diff, but there's been a real focus on utilizing these therapies in ulcerative colitis as well. And there has been some real early success specifically in that space. Now, FMT is like a broad shotgun approach. We're now really seeing more precision-based microbiome approaches where we can really try to focus in on certain specific pathways such as butyrate production, increasing short-chain fatty acids, which of course are critical for inflammatory pathways. And so this is a real big area of development you're going to see in the next few years with some really important trials underway right now.
Okay, that's good to know. So maybe I wanted to ask the three of you, how you think about risk reward for each of these different modalities. So maybe we can do this in reverse order this time. We'll start with Dr. Allegretti on your views about Tregs and microbiome and what you think the potential areas of concern might be, or is it too early to know?
Yeah, I think that's an excellent question. And I think too, often when we're finding these patients who are going to be enrolling in some of these trials, even very early phase, again, we're looking at patients who really have very poor quality of life, who are up against potentially needing, life-changing colectomy or really morbid surgery. And so again, when you think about risk, reward, benefits of potentially wanting to trial some of these therapies in this population. This is actually quite an eager patient population because they're already quite miserable with a fairly morbid disease. And I think the nice thing about the pathways that we've just discussed, specifically low-dose IL-2 as well as the microbiome, we're already really seeing excellent safety profiles. So as opposed to really suppressing the immune system where you do worry about severe infections and even cancer formation in certain populations, when we think about stimulating the immune system, we really don't see as many of those risks. And again, with the microbiome, I think, again, there's still a lot being studied, but we have now years and years of experience with these therapies in the C. diff space. We're even enrolling, in a 10-year national registry now, and we're really not seeing the initial safety concerns that were originally proposed.
Okay. Are there certain types of conditions that you think are particularly amenable to microbiome treatment?
That is a great question, and I think even within IBD, like all of our therapies, we don't have 100% efficacy with any IBD therapy. On average, you get about 30% remission given on which mechanism of action you're speaking to. So I certainly believe that there will be a subpopulation within both ulcerative colitis and Crohn's that are going to be essentially microbiome mediated, and those are the people that will really benefit from therapies like this. So far in our experience, it does seem to be people who have more mild disease. Some of the early clinical trials were really focused on those with end-stage disease, and that's clearly not where these therapies are going to function at their best. So earlier disease, think mesalamine failures, almost early biologic, and I view these as potentially steroid-sparing agents as well, where you could quickly induce someone and hopefully avoid steroid side effect.
Okay. Same question to you, Dr. Mempel, about how you think about the potential risk-reward profiles.
Yeah, so obviously the cellular therapies can build on the experience of cellular therapy in the cancer immunology, cancer immunotherapy space, and a lot of experience has been collected there. The biggest safety concerns are probably for genetically engineered cell products, insertional mutagenesis through the use of integrating vectors, retroviral, lentiviral vectors, transposons. And of course, there the safety equation is a very different one in the context of autoinflammatory disease, which are generally not life-threatening. So here for the patient, the threshold must be much higher to achieve really an acceptable safety profile. However, there is also really truly debilitating forms of autoinflammatory diseases. And I think in these instances, I think the patients would actually be accepting, and we should be willing to provide these kinds of therapies. And that is being done with considerable success so far, really amazing response rates in patients that had no other treatment options and also life-threatening disease. So I think we are basically gaining ground in that area. The insertion mutagenesis risk can potentially also be addressed in the future using non-integrating gene delivery methods, for instance, episomally replicating DNA vectors are one option there that basically don't dilute out like other non-integrating vectors, but that basically replicate episomally in dividing cells and therefore can also give long-lasting gene delivery without the risk of insertional mutagenesis. So those vectors are now basically maturing. They used to have viral components, and those have been engineered out, so they have also lost risk associated with that. The other, apart from insertional mutagenesis, the general risk of, for instance, CAR T-cell therapy are cytokine storm and neurotoxicity, also prolonged cytopenia, B-cell aplasia. Those, for reasons that are, I think, not fully understood yet, seem to be much milder when CAR T-cells are being used to ablate B-cells for autoimmune therapy. in patients. So the rates of neurotoxicity are basically in single digits, if at all, and cytokine storms are very rarely observed as opposed to cancer patients. So I think the general safety profile is much better there.
What do you think about that point that you made on B-cell depletion, the thought about consistently depleting B-cells for certain types of treatments? How do you think about that in terms of longer-term safety, especially for autoimmune?
Yes. So, I mean, right now the strategy being used is general B-cell depletion. I think in the long term there will be refined methods to deplete selectively pathogenic B-cells. For that, we would probably need to identify autoantigens more precisely in order to be able to target specifically the pathogenic B-cell populations, because obviously long-term B-cell depletion has its own inherent risks and also in context of infections.
As it sits today, do you think that this methodology would be applicable to all of your patients, or would there be like a subset of patients that you would recommend this type of treatment to?
I think right now we should stay with the patient populations of really with debilitating and life-threatening disease until we gain more experience and until also cellular engineering, genetic engineering methodologies have matured further.
Okay, got it. And then Dr. Lester, same question to you about complement and FC.
Anytime you're inhibiting the immune system, you're going to invoke the possibility of infectious complications, because that's how the immune system evolved, to deal with pathogens. So it's sort of a double-edged sword. And we know, like for complement deficiencies, patients are susceptible to encapsulated bacteria. So you're always weighing the risk-benefit. But the clinical trial that got avacopan approved was steroid sparing. So the complement pathway inhibition was thought to be less toxic than chronic steroids. You're weighing the risk and the benefits there. And the same thing with the FC receptor, neonatal FC receptor blockade. I mean, there, you're lowering immunoglobulin levels. So as opposed to like antibody therapy against B cells like rituximab, or CAR T cell therapy that will even further wipe out the B cells, the safety profile might be better for these agents in that you are not affecting the B cell compartment, really, you're just affecting the antibody levels. But having said that, you're lowering all antibody levels, not just the pathogenic antibodies. So our antibodies that we have do lots of things that are good, fighting off infections and things like that. But can you lower the pathogenic ones without affecting the protective ones? I think time will tell, but it seems to be encouraging. But I think the holy grail for immune therapy is to identify those clones, those T cell clones, or those B cell clones, or those antibodies that are really pathogenic, and then wipe those out, as opposed to wiping out all B cells, all T cells, or the whole, all antibodies. So if you could target particular T cell clones or B cell clones, if you knew the autoantigen and have antigen-specific therapy, I think that's the next, sort of the holy grail of immune therapy. And things like CAR T cells may actually get us there, as opposed to engineering those cells to wipe out all B cells. You can engineer them to wipe out only B cells that express a particular antigen receptor, or even target against T cells that express a particular TCR once they've been identified. And the same thing could be true for regulatory T cells. If you could engineer the regulatory T cell with a T cell receptor or a chimeric B cell receptor that will target antigen-specific cells, this way you could selectively inhibit antigen-specific cells in the immune system, then you have a much better immunosuppressive drug that's very specific for instance, clones of T cells and B cells that are making antibodies against the acetylcholine receptor. That'll eliminate myasthenia gravis without affecting any other antibodies or T cells or B cells in the body. So I think that's sort of the next phase of immunotherapy will be antigen-specific therapy.
Maybe Andy and Thorsten, I have a question for you. So a lot of our conditions, IBD included, but especially in rheumatology, the antibodies are associated with the disease, but we don't know what the antigen is. And so you bring up a good point. You know, we hesitate with B-cell therapy because patients are at high risk for infection, especially in the era of COVID. Is there a way, how do you see, let's just use rheumatoid arthritis as an example, could we get there if we don't know the antigen?
Yeah, so once you clone out with single cell sequencing, you can get a cell, a T cell or a B cell, and you could actually clone out the T cell receptor or the B cell receptor, so you know what those T cells or B cells are seeing, and people make a guess that T cell receptors that are expanded in the joint in patients with RA, that they're probably reacting to something in the joint and driving the disease. They're not in the blood, they're in the joint, they're expanded. Now you can clone out those T cell receptor, and actually there are methods now for both class 1 and class 2 antigens to actually identify the antigen. And actually someone at the Brigham and Harvard Medical School, Steve Elledge, they just did that did a T-scan 2, they call it, for identifying antigens in Sjogren's patients. By actually cloning out the T cell receptors and identified 2 antigens that are expressed in the tissue in those diseases. Now, having said that, it's not always that simple. So a lot of the T cell receptors are picking up antigens that people are scratching their head at. What does that have to do with the disease? It may not be in the disease tissue. So there's a lot to learn, but the technology's there now to clone out T cell and B cells that are probably pathogenic. They're in those patients, they're enriched in the disease, and then figure out what those antigens are, and then maybe go backwards to inhibit those pathways.
Great.
Yeah, maybe just to add to that on the subject of regulatory T cells. I mean, one beautiful aspect of regulatory T cells is that they adjust the level of immune suppression that they provide to the inflammatory context. So basically their function, the clonal expansion, is linked to cytokines produced by effector cells, such as, for instance, IL-2, produced by effector T cells, pathogenic effector T cells, for instance, but also to the degree of co-stimulation that they receive in inflammatory environments. So as soon as inflammation dies down, regulatory T cell activity also dies down. So basically there's the self-regulating aspects to regulatory T cells. So I think there's less of a risk to generate generalized immune suppression with a regulatory T cell, especially if it's possible to target the regulatory T cells to self-antigens that are expressed in diseased tissue. And it doesn't even have to be the autoimmune antigen. It can be actually any antigen that is expressed in that particular tissue that can be targeted by regulatory T cells using TCR engineering or a CAR, because this will focus the activity of the regulatory T cells on that tissue where the antigen is expressed and will not generate generalized immune suppression everywhere else and thereby run the risk of infection. So it's a very targeted immune suppressive opportunity.
Yeah, that's very exciting. So we don't need to know what the causal antigen is anymore. You just need to know what the cells are targeting, right?
Yeah.
It's very exciting. So maybe we'll pivot a little bit and talk about acute and chronic and how you might think about these targets in the setting of acute versus chronic inflammation. Jessica, maybe we'll start with you.
Yeah, I think the sort of corollary to that in the IBD space is induction and maintenance, right? So we think about acute inflammation, this acute event where the patient is sick, inflamed, having active symptoms, and you need sort of something immediately to sort of put that fire out versus something that's going to long-term keep under control this chronic inflammatory condition. So we have both therapies that treat the acute inflammatory state, induce remission, and then therapies that maintain remission. So the sort of classic strategy for acute inflammation in our space is steroids. And of course, we know that of all the things we're discussing, steroids by far and away have the highest risk and the most sort of side effect profile. And so when we think about some of these novel pathways, you think about where they play best. And so as we were discussing, the novel microbiome therapeutics may be best suited to serve some of that acute inflammation and actually help induce remission quickly but they may not serve well for maintenance therapy and have somebody on them long term, whereas then we can kick in with some of the more immune-based therapies that's really going to be used in the maintenance space. And so when we think about novel drug pathways, sort of, are you putting out the fire immediately, or is this something that patients are going to be on long term, because these are lifelong diseases?
That's great. And Thorsten, what do you think about with the Tregs? Is there a role for acute versus chronic, one or the other?
Yeah, I mean, so I'm not a clinician, so I don't have the experience of treating acute versus chronic patients, but bursts of inflammation. But I don't see why also in the acute setting, a regulatory T cell could not be effective. Of course, the limitation is the manufacturing time. So the standard right now is autologous, cell therapy, and there you need to harvest the cells in a suitable state. You need to process them, expand them, and the time that it requires for that is probably longer than you would want if you have an acute inflammatory event that you have to immediately address. So I think that will be the limitation at the moment. If there's other engineering methods that maybe, for instance, in situ gene editing of cells, those could actually provide for a more rapid, treatment modality that utilizes regular T cells, and that then could potentially also be useful in an acute stage for disease.
Right, so what I'm hearing is, like, feasibility aside, if you're able to do it quickly, it might be something that you could start right away with the steroids and get the steroids off quicker, which is what we're always looking for. That's very exciting.
Right, yeah. And I mean, IL-2 therapy that was already mentioned, that's something that's available off the shelf, essentially, and it could also be implemented much more rapidly. Definitely. But then, of course, in the acute settings, effector cells also express IL-2 receptors. Yeah. So as was already mentioned, then you want to have to very carefully balance the dose of the IL-2 mutant.
Definitely. So Andy, what are your thoughts about complements in these
I
settings?
think, just to follow up on Jessica's point, I think when someone comes in and is and they're presenting with their disease in the acute setting, you're going to want to use immunosuppressants that can really get control of the inflammation quickly and limit disease pathology. So you're going to really use a sledgehammer, if you will, give them steroids or give patients something that's going to really get the inflammation under control, because you may start out with one pathway getting started, but if it's a complement pathway and you're recruiting in neutrophils or eosinophils or other cell types that are now releasing all their mediators, you want to quiet those cells and all their products quickly as possible, and then in the chronic phase, go to the more really targeted therapy. If we understand what is the disease pathway that's initiating the process, so a good example might be somebody who's has a big asthma flare and they're getting steroids because you want to suppress the entire immune system, including eosinophils and neutrophils, but then you want to get them onto something like dupilumab, where you actually inhibiting IL-4 and 13 pathway only, and you're not really affecting neutrophils or really eosinophils directly. So then it's much more targeted in the maintenance phase. So I think early on you're not so elegant because you want to get inflammation under control and limit organ damage. But then in the chronic phase, when people are more chronic, you want to have the most targeted therapy possible to get the disease pathway that's initiating it and having the least side effects.
Yeah, I guess one thing that I was thinking is, as all of you were speaking, is we think about acute and chronic, but you're really also thinking of early versus where the horse has left the barn, right? So it could be that if you catch it before it sets a 5-alarm fire, right, you could use these more targeted therapies. But after that, you just got to blast them with steroids and then get it under control and then go to the targeted therapy.
It is interesting because even some of our sort of newer agents that are coming to market now, I think sort of the real focus is speed, right? Everyone wants to know is how fast, when do patients start feeling symptom relief again? Because if you have therapies that are a bit more targeted but do work extremely fast, then you may be able to skip that initial step. And so I do think this is sort of where the field is moving towards. How fast are these agents working? How fast can you get them into the patient? And can you bypass that sometimes necessary step of oral prednisone or IV steroids?
Right, and I think some of that might be also access, finding the patient early and having them be able to get in early.
Absolutely.
Too.
And getting the prior auth, of course.
Yes.
So maybe a couple of follow-ups from me on what we've just discussed. Maybe the first one for Dr. Lester. How are you thinking about different targets within the complement cascade? In the world where I reside, we talk to drug companies all the time that are trying to find new ways to treat diseases, even if there are already other treatments available. So can you talk about the difference of classical versus alternative pathway, why it is or isn't important to be targeting something that can be inclusive or exclusive of both?
Yeah, so I mean, the alternative pathway I think is grown, let's take a step back. The classical pathway is thought to be activated by when antibodies activate the complement pathway and activate a particular set of molecules that lead to the membrane attack complex. The alternative pathway is a more subtle pathway that can get activated by many things. It can be activated by antibodies, but can also be activated by other things in tissue, and that can tick over and activate the complement pathway. It all comes down to the same spot eventually, generating C3 and C5a and the membrane attack complex. But I think there are sort of different strategies, because you can have, a ligand inhibiting like what's used in PNH, where you're inhibiting, complement itself, and then complement itself generates C3, C5, and then those hit their own receptors, and there are 2 receptors for C5a, and there's 1 receptor for complement. So you could target particular receptors, which is being done now. That's like avacopan, which targets C5AR1, and it leaves C5AR2 completely intact, and the biology of that isn't quite clear yet, if that's pro or anti-inflammatory. That's still being worked out, but you can really target C5AR1, where you inhibit complement upstream. You could be inhibiting the generation of C3 and C5, or you can inhibit C5 and not affect C3 and C3a and the C3a receptor. So, there are a lot of nuances on, if you want to, what you really want to accomplish in inhibiting the complement cascade. So, like for instance, for PNH, you don't think you'd want to attack C5AR1 because that's not what's actually causing the damage. It's the activation of complement on the surface of the red blood cell. But for ANCA-vasculitis, the target was really the neutrophil complement receptor, C5AR1. So I think there, if you know that's the target, you want to be as specific as possible, because actually we don't really understand the biology of C5AR2, so why block that? Because it may actually be yin-yang and actually be hurting you. So it's a question of being how specific you want to be and how broad you want to be.
Yeah, and on that point, maybe going back to FcRn as well. So for certain conditions like myasthenia gravis, it seems like the pathogenic autoantibody is already known just by results we've seen with efgartigimod, which is an anti-FcRn. But what about where you're not really clear what the source of the antibody, the original autoantibody is, and there might be other autoantibodies that are associated, right, that does it make sense to target those?
Yeah, I think that's really interesting. I'm really looking forward to seeing how those trials pan out, because as you alluded to, and I think Kat alluded to, that a lot of the, particularly the rheumatologic diseases, are associated with autoantibodies. That's how diagnoses are made, but actually, we don't really fully understand how pathogenic they are. Diseases like myasthenia, pemphigus vulgaris, where there's an antibody to one of the desmogleins that lead to the disease. There, it's clear there's a pathogenic antibody. But for things like even rheumatoid arthritis or lupus or Sjogren's or a whole host of other rheumatic diseases, they're associated with particular autoantibodies, but we don't actually know the pathogenesis of those antibodies. So it'll be interesting to see if you lower them, if actually you could target the disease. And I think it's an open question. I think it'll be quite interesting to look at that.
Okay, and maybe one more question for me for Dr. Allegretti as it relates to the microbiome. I think in the past there were a lot of questions because in the initial days of microbiome it was all about needing donors, but now it's moved at least in part to synthetics. Do you have a view on where it's important to have live donors versus synthetic development of microbiome treatments?
Yeah, I certainly hope that in the near future we're not using any live donor-based therapies, not because of safety, but really because you can imagine it's quite difficult to source that material, and it really does make production of these therapies quite difficult, which I think is partly why maintenance-based therapies using these has really been poorly studied, because again, that's a lot of donor material that needs to be sourced. And as we have emerging pathogens like SARS-CoV-2, like monkeypox, I mean, you have to be able to be very nimble to be able to screen for these things in real time. So you certainly have more control as we move to more a synthetic lab-based product. Now, I think the initial issues have been that in the earlier days, things that were lab-based or synthetic just did not perform as well as donor-derived products, and it was partly because nobody really fully understood the exact mechanism as to how many of these therapies were working in various disease states. It clearly wasn't just the bacteria, but probably a combination of the viruses, phages, metabolites, everything that was comprised of whole stool. I think the science has certainly come quite a way, and we are able to now engineer clusters of bacteria, and so you're not seeing usually single strain products, it's usually multi-strain products, and we have some of the first phase 3 trials right now from Vedanta Biosciences looking at the prevention of recurrent C. diff, which will be the first synthetic product if it does come to market, and I think once we have that, we'll start to see a lot more development in several other inflammatory conditions.
Great, well, we have a few minutes left, and we'd like to open it up to the audience to see if anybody else has questions before we keep asking more questions. There's someone in the front.
You mentioned about the interleukin-2 trials. Is there any early clinical proof of concept already for IBD?
Yes, so we actually did the first low-dose IL-2 trial in ulcerative colitis that is published. This was a dose-finding study where we actually used 3 low doses, and we actually found that there was a dosing threshold that actually, at a certain dose, you actually do start to upregulate conventional T cells, and that really increases the side effect profile that the patients experience. And so we have completed the proof of concept in ulcerative colitis and are actually enrolling right now in the Crohn's trial.
Over there?
Hi, I wanted to know a little bit more about, you spoke about avacopan being used for different autoimmune diseases. Can you speak a little bit more about that, if you know about potentially clinical trials where it's being utilized for different diseases?
Yeah, I actually don't know. I mean, from my understanding is Amgen is looking to extend the use of avacopan in ankylosing, but I'm not sure they're doing a lot of trials in other diseases. There are other complement inhibitors that are out there that are being tried in other diseases. Yeah, that's I can't name them actually at the moment, but there are definitely others out there. Yeah.
Any other questions?
I have a question regarding the heterogeneity you're seeing in inflammatory diseases. So I'm a clinical researcher. I have done a lot of studies in various diseases, asthma, UC, and so on. And I was always interested looking at the bell-shaped curve and looking at the patients on the left and in the right and the tail end. And I was so impressed by the fact that how individually they reacted. So the so-called average patient in the middle of the bell-shaped curve doesn't exist. And if you think about the immune system, that's what you would expect, that there's a lot of heterogeneity. So when you're doing your clinical research, you're looking for finding an answer whether something works or not. How do you take heterogeneity into account?
Yeah, I mean, heterogeneity in clinical trials is a huge problem. And I mean, that's partly why we randomize, right? Randomization is supposed to account for at least part of that. And I think that's why you'll see, I think notably, especially in the IBD space, many clinical programs go for ulcerative colitis first. And it's not because there is an expectation that many of these drugs or pathways will perform better in ulcerative colitis. It's that in itself is a much more homogeneous disease than Crohn's is. Crohn's is probably 1,000 disease in one disease, and so when you're designing Crohn's trials, you have to really decide very specifically which phenotype you're going to be specifically targeting, just making those trials a bit more complicated, whereas ulcerative colitis, you do have a bit more even playing field, and so I think as you're seeing drug development in the IBD space anyway, you often see programs going for ulcerative colitis first, and that's sort of one way to sort of assess proof of concept in a more homogeneous population.
I think that's a real challenge, though, is because, some diseases, the clinical manifestations are the same, but they can be driven by different pathways. And you have to design that if you're going to target the pathway, you have to have those patients in the trial, not everybody. I mean, the asthma therapies were a great example of that, where until they, did the clinical trials for the IL-5 eosinophilic high patients, the IL-4 and 5 therapies or 13 therapies didn't work. But when they did work. So I think, profiling the patients and being able to segregate them in ways that reflect maybe the critical disease-driving pathways, I think will lead to more, better trials and more approvals for drugs. But it's an important point.
And I think to that point, though, you're going to start to see a lot of new programs coming out with specifically companion diagnostics. I think to that point, whereas we're starting to see a lot of novel pathways with biomarkers on entry. So we're starting to see, because to your point, many of these diseases are driven by different pathways, even though they have the same clinical presentation. So if you can enrich your clinical trials with people specifically whose disease is being driven by the pathways, obviously those trials are going to be better done, you're going to have more power, et cetera. And so I think the new wave, at least in IBD, is these companion diagnostics for really better targeting.
And I might just add something for rheumatoid arthritis. It's clinically has a definition, but we find it's a very heterogeneous disease. So there is this effort now to look at the tails, and through, some of you may know of the Accelerated Medicines Partnership, where they're actually going in, looking at the synovial tissue, and trying to kind of re-phenotype these patients to understand who's in the extreme.
I have maybe one question also. For Dr. Mempel, you talked about the importance of making sure that you have the right population identified for cell therapies, but what about the targets themselves? It seems like a lot of companies are targeting antibodies to CD19 right now to look at various inflammation diseases. What's your view about what the threshold would need to be, let's say, on, IgG knockdown, for example, for a cell therapy versus one that's potentially less harsh on the patient?
Yeah, I mean, I'm not a clinician, as I mentioned, so I don't really have a good sense of patient presentations and to make a decision.
But I guess theoretically on what you'd like.
I think, as mentioned, CD19 is a crude target, or BCMA or other, CD20 B-cell targets. I think the future will be to actually, target selectively the pathogenic, B-cell populations. And I think that will lower the threshold for intervention, clearly, given if the safety of the genetic engineering can be addressed, and will probably make those therapies more broadly applicable, because they have the potential to be curative, and just so as regulatory T-cell therapies, or at least provide very sustained responses to patients. So, which is very attractive, obviously.
I mean, many of the diseases, entry criteria, they have to fail standard therapy.
Yes.
So you're getting a population that's refractory and more severe, and the patients have to agree to the risks. But they're usually patients that have failed other therapies, if it's therapy with more serious side effects.
Do you see a world where cell therapies could be frontline treatment? And
could be what?
Could be first-line treatment ever? And what would need to happen for that?
I mean, the threshold for that is manufacturing cost at this point.
Yes.
which will likely come down. And as I mentioned already, in situ engineering of cells, genetic engineering of cells in vitro will also provide things more quick and more cost effective. And then, yeah, the safety aspect, that's I think the critical element that will broaden really the patient range. It could make it a first-line therapy for certain diseases, yes.
If you have cell therapies that are off the shelf, more allogeneic than autologous, that would make the costs come down. But the cost is going to be an issue.
Yep.
Yeah, for instance,
and then the side effects as we see what happens over time.
Yeah, I mean, for instance, in the allo transplantation, in the hematopoietic stem cell transplantation setting, we've now seen enormous benefits of regulatory T-cell therapy by bringing down graft versus host disease from rates of, let's say, 40% of patients to rates of 3 to 4% of patients. I mean, that's an enormous benefit. And I think in that setting, in that hematopoietic stem cell transplantation for malignant diseases, I think in that setting, there's a short path to making this a first-line therapy. Yes.
Perfect. I think with that, we're out of time. Yeah. So thanks, everybody, for joining us on the panel this afternoon. Thanks, everybody, for joining, and we look forward to continuing the conversation at future times.